right, so we should get started. It's my great pleasure to introduce today's speaker, Srini Charaga. Srini's been here a number of times, but he's one of the few speakers I know whom it's not. You can never get too much, right? So almost every year he comes, and almost every year he says something new to tell us and teach us about neuroscience. So Srini was trained mostly in neuroscience in Cambridge. He was a PhD student down the street with Sebastian Sung, and did a postdoc in London before becoming a group leader at Janelia, where he's been for the last several years. It's sort of hard to introduce Srini and all this he has done, because he has worked on such an enormous range of topics in neuroscience, from connectomics to whole brain imaging to computational modeling of large circuits and understanding behavior and so on. So just to give a flavor of Srini, so I'm going to probably irritate a number of people in the audience, but I sort of view theoretical computational neuroscience as sort of two types. All right, those people who sort of revel in data, I think that's Srini, and those people who recoil from data. And maybe it's overstating it, I don't know, but data makes theory hard, right? Because data right? Because you have a beautiful theory which is sullied by too much data, right? You have your beautiful theory which fits in a nice Tupperware box, and you start loading it with data. Eventually there's too much, and it starts hard to close the box, and eventually stuff starts spilling out, and either you throw away the box and get a new box, or I don't know, the metaphor starts to break down. But Srini is one of those people who really he sort of epitomizes the data-driven approach to neuroscience, and I think before I irritate too many more people, I should sit down and let him see what he has to tell you today. Okay.
Thanks, Harvey. Those of you who want to stay after all that, thank you. And thanks also for coming. Do you have a preference for which screen I point at? Okay, I'll point at this one. I think I like standing in front of the screen. Okay, so what I'll talk about here is, as Harvey pointed out, using data to constrain and derive mechanistic models of neural computation. So what does that mean? We've got a brain, and those of us who are here are interested in trying to understand how this brain computes, and a lot of what it does is to turn stimuli into behavior. This is sort of the inputs and outputs to this block, and this black box contains neurons, and as neuroscientists, we'd like to understand how the brain uses neurons to solve these computational problems. So in my work, I'm interested in trying to figure out what is the neural network. What's the structure of the particular way in which the brain solves these computational problems, to as near as we can get it. Can we identify the precise neural network that solves these problems? And in particular, from a mechanistic point of view, what is the role of individual neurons, cell types, connections, brain regions, these things that as neuroscientists we can measure? What is the role of these things in the computation that we're interested in? And if you have an understanding at that level, that can teach us what the effects of causal manipulations are. It can teach us, if I perturb these neurons, or maybe knock out those neurons, or if I have some sort of disorder, what happens? And in order to make predictions at that level, you need to really understand how the brain actually solves these problems at some level of detail that has to do with what. And in the future, you'd like to think that this can also, this sort of level of understanding can also teach you something about why the brain sort of was evolved to solve the problem in this particular way. I'm not really going to answer that question, but the hope is that this sort of process of trying to identify the precise way in which the brain is solving certain problems will try to get us there. Okay, so the actual things I'll talk about is, the fruit fly visual system, where we now have a lot of the data that I'd like for this, which is in the form of connectomes at the cellular resolution. You know, we'll try to build a model over there. And the second bit is in bigger systems, like a rodent, trying to understand, you know, how single neurons compute, and then how the network of neurons in local pieces of brain tissue are wired. So that's sort of the general outline for the talk. First thing first, you know, there's a lot of work now in recent years from various people, including David Cox, who used to be here, Jim DiCarlo down the street, and so on, where they tried to open up this black box by putting in constraints into this neural network and then training it to perform tasks that we know that we are capable of. So if you're interested in studying the primate visual system, one task that you might imagine that the primate visual system does is recognizing objects within images. So what you might imagine doing is to build a neural network that has similar structure to the way the primate visual pathways are organized. And now we use this new technique we now have available called deep learning to train this network with these constraints to solve particular tasks. This is something people do in computer vision all the time now. So can we take those models and then, which originally were themselves inspired by the brain, and start to make more detailed connections to physiology? And so from our view, you know, here's a convolutional neural network model of the style that people use in computer vision for doing object recognition, which might take an image, detect local features, and then categorize it into different sort of categories. These stages are meant to correspond to different stages in the visual pathway. These are different brain regions, primary visual cortex, V2, V4, and so on, until you get to IT cortex, which is where people believe there's, you know, an object code. And so what these people were able to do was to find that there was a correspondence between the features that neurons in these brain regions responded to, between those brain regions and layers within this neural network. And this correspondence emerged because of the constraints they put into this neural network to have this particular layered architecture with particular types of connectivity. So, you know, we can get now predictions at the level of brain regions and populations of neurons. Can we make more detailed mechanistic predictions? For that, we'll need more constraints. So that's sort of the idea that we're going to go at. And we're going to look at the fruit fly visual system. The fruit fly has two compound eyes with about 700 pixels each. And those 700 pixels are arranged, they're called ommatidia in this hexagonal lattice, and each ommatidium has about 8 photoreceptors in it. And then there's some stages of processing in this fruit fly retina. And so we can try to understand if we can try to make, we can try to use recent data that we have in the circuit to make models of this system with much more constraints than people have been able to do in the primate visual system. And that's because we now have access to connectomic data information, which tells you for individual neurons how they're connected to other neurons in the circuit. That's not something we have for the primate visual system. We can only have that here because the system is small, the fruit fly itself is small. And so the way you go about this is you do electron microscopy, and then you trace the neurons in there, and then you find out which neurons are connected to which, how many synapses they make with each other. And this is information you can measure from 3D volumetric sort of electron microscopy by doing image analysis there. And that tells us, you know, it gives us the detailed morphologies and it gives us the counts, but there's still a lot of missing information. So imagine that you want to simulate this and sort of try to make predictions about what's going on. There's still quite a bit of information that's missing. In particular, we don't know the single neuron biophysics. We don't know the input-output functions of these neurons. And that's something we need to put in to our model in order to simulate this. We also don't know how to translate the measurements that we do have, which are synapse counts, into synaptic strengths and time constants and so on. So So even the simplest types of models that we'd like to be able to make, involve information that we don't yet have. But there is quite a bit that we now do have, which is, the precise nature of which neurons are connected to which is a lot more information than we had in the past. So now we have to figure out what's the scale at which to bring this sort of information we got from a dead brain to life. So I was interested in this from the perspective of, a machine learning person sort of theory. From a theoretical perspective, what we've known for a long time is that neural networks, if they have enough hidden units, multi-layer neural networks, can be universal functional approximators. They're really powerful in that they can compute any function you throw at it, which is nice. That means that we can use them to solve various machine learning problems if we have data. What that also tells us, a corollary to this, which becomes obvious if you sort of think about this, is that if I know what computation a particular neural network is performing, it doesn't uniquely constrain the particular form of the neural network, because I can compute the same function in many, many different ways by adding more hidden layers, by adding more hidden units in each layer, and so on. And we also know this from theoretical neuroscience. So for many computations that people describe in the brain, we have many models that theorists have come up with. And we don't yet have enough data to constrain and figure out which of those models is accurate. And so when you sit down and think really hard and try to think about the optimal network for computing a particular function, we found that there's degeneracy there. So knowing the computation doesn't tell you how it's performed. And there's many solutions. But the converse might actually be true. So a neural network that's constrained to only connect to certain neurons may not be able to compute any possible function. And so what that means is that it may be hard to predict structure, the particular form of a neural network that you might use to perform a computation, from the function itself. But it may be easier to predict function from structure. And so this is the thing that we want to test in this context of connectomics and how to use this sort of new connectomic data. I should say that at any point, if you have questions, do shout them out. Raise your hand, and we can talk. So please feel free to ask questions. Okay, so here's a sort of toy example that illustrates this point. So imagine that I take two random networks. Network 1 and network 2, they have completely random weights. They're different. And then random thresholds at which they'll fire. They're called biases. Now if I take neuron 1 in this network and neuron 1 in the other network, there's no reason that there should be correspondence in any way. What I can do is I can feed both of these neural networks the same patterns of input. And what I can ask is, what does the firing pattern of this neuron look like versus the firing pattern of the other neuron? I can quantify this. What I'm showing you here is how correlated they are. These two networks are receiving exactly the same stream of input pattern, the same stimuli, but they compute and fire in wildly different ways. So if I look at the correlation, how correlated they are, the first layer, all the layers, the correlation is 0. But the first layer, has some spread to it, which means that some networks are maybe more correlated than others. If I do this average over lots of networks. But on average, they're just not very correlated to each other. So random networks compute uncorrelated functions as you sort of go through this neural network. But now, let's add in some constraints. Let's add in constraints which tell us which neurons are connected to which. In the past, I allowed all neurons to connect to each other in this feed-forward network. Now I'm going to make them very sparse, which is similar to the way that that biological neural networks are. Not all neurons connect to each other. And then, we also know something about the signs of these synapses. We can measure, whether a neuron is excitatory or inhibitory, and I can put that in. So now the question is, this is from the perspective of someone who has a connectome, which tells you which connections exist and which ones don't, and who also knows separately what the signs of these synapses are. If I simulate random networks with these constraints, how different will they be? So I do the same experiment as before, and now what I find is that in the first layer, these neurons, the same two neurons, are much more correlated than they were in the completely random network. And so what that means is that these functions are much more similar to each other. It also means that if I pick one of these functions, it's not going to be that far off from the true function that's being calculated. Yeah. Yeah, so I played around with that to try to sort of, make this as bad as possible, and so that's what this is chosen to be. But it'd be fun to chat about what that number should be if you can calculate it. I don't know how to calculate that yet, but it's chosen not to be easy. Okay, so what you now find is that in the very first layer, two networks that have the same stream of inputs compute very similar functions. But as you go through the network, it starts to diverge. They start to compute. These differences start to add up, and now you can start to compute, different functions from each other. Yes?
Can you just flesh out what constrained sign and sparsity means?
Yeah, so what that means is that both of these networks have exactly the same neurons that connect to each other, and exactly the same neurons, pairs of neurons, don't connect to each other. So the zeros and the non-zeros in the weight matrix are the same. The signs of those synapses are also the same. So that's the constraints. Other than that, the strength of these synapses is allowed to be different, as is the threshold at which the neuron fires. Is that? Yeah, great. Okay, so, but now, one thing I didn't do is I didn't constrain these networks to perform the same computation. If they were constrained to perform the same computation, at the last layer, they should also be very similar to each other. If they're all doing object recognition, then they should all perform exactly, to give you the same sort of object classification at the end. So constraints at both ends can sort of help. Constraints combined with some constraint about what the computation is should help you sort of make these two networks very similar to each other. So constrained networks can compute similar functions. Just a summary, random networks with constrained connectivity compute similar functions in the early layers. Trained networks will compute similar functions at both the early and the last layers. So now what we want to use is to use these connectomic constraints that we get from measurements that we can now make, and work in a place where we know what that function should be. And know as sort of in a loose term. We're going to be relatively flexible about what we mean by knowing the function. We're going to just say we know for the fruit fly visual system that it does vision. That's about it. Not much more than that. Okay, so people at Janelia, where I've been working for the last few years, have mapped the connectivity in this fruit fly optic lobe over several papers. These papers that I'm pointing out here have looked at the first two stages of computation, the stage that's called the lamina and then the medulla. There's now work further downstream, but this is what I will focus on for now. Just to refresh your mind, so there's this retina which computes where the photoreceptors are, and then it has this hexagonal lattice structure, so the pixels of this camera sit on this hexagonal lattice, and then under each of these cells is a column of neurons that perform computation. So the lamina has the same columnar organization as the ommatidia and the retina, and then the medulla and so on. So that means that there's actually a strong analogy between the circuitry there and a convolutional neural net. So if I sort of simplify the wiring, what I have are these 8 types of photoreceptors, which are connected to these sort of lamina neurons, which appear once per column. So in the fruit fly visual system, they all have names because this circuit is really stereotyped from animal to animal. There isn't a lot of plasticity in this circuit. So, you know, individual neurons have names, and we now know which ones are connected to which, and that's what this diagram shows you. And so these are all different neurons which appear once per column. So, yes. So this is sort of averaged across all columns. So this is telling you which cell types are connected to which cell types if I just look at the entire eye. This is what tells you here's the entire collection of neurons of that type. And we made this diagram and we thought we could stare at it and it would be useful to make this diagram. I'm not sure that it necessarily is, but this tells you, you know, for that particular neuron who it projects to, and so sort of in a spatial sense. It's just a couple of columns. So it's 7 columns, so a center column and a couple of surrounding columns. And so this is the representation, yeah. So this is the real representation that we have, which is for a particular neuron in the center column, how many inputs does it receive? How many synapses does it make receive from a neuron of this cell type in its own column, and how many does it receive from columns that are around? And so, you know, this is the reconstructed data that we have. So it's a local reconstruction that tells you what the spatial profile of synaptic inputs are. And so if you are familiar with convolutional networks, this is the filter of your convolutional network. And this is which feature maps connect to each other. So the sparsity pattern at the level of feature maps, where in reality, each of these circles is expanded out to be 700 neurons, or a feature map. So these are the feature maps, and those feature maps are connected as follows with these filters. Is that sort of clear? Does that make sense?
Can I ask a very quick question?
Yeah.
So just for historical purposes, is it fair to say that the general connectivity is known and you put sort of numbers to how many synapses so on, or new connections were discovered?
Certainly new connections were discovered in the process. I think new connections were discovered, but the coolest thing that came out of this was a spatial profile of those connections, because that is something that you couldn't have gotten very easily in other ways. So yeah, that's the detail that you needed this technique to power. Yeah. So this network has loops, and we model all of them. Yeah, so this is modeled as a recurrent neural network. Okay, so here's how we're going to model the circuit. We're going to model it as similar to a convolutional neural network, but it's not exactly that. It's sort of a hybrid of computational neuroscience model, which is called the threshold linear neuron, which is basically a neuron that has a time constant and has an activation function that's threshold linear. So it means that, you know, below a certain threshold, there's no activity, and above it, there's activity that's proportional to the strength of the inputs. Now, these neurons don't spike in these early stages of the fruit fly visual system. And so this model, which is in computational neuroscience called a rate model, because there's no action potentials, it just has this continuous activation, is actually the right model for the circuit because these neurons don't actually spike. And then we had instantaneous synapses, and so the main things that set the time constants, the filtering properties of these neurons, is a single cell property, so the single cell dynamics. And then you know, we get the signs of these synapses from other experiments that people have done, also on dead brains, which is, you know, RNA sequencing that you can do, so molecular profiling, to figure out which neurotransmitters and which receptors are expressed, as well as fluorescence in situ hybridization experiments. So in this class of models, with these point neurons, with these continuous activation, there's two parameters that are missing, and those correspond to the single neuron properties. They're the threshold for each neuron and the time constant, so how much does it remember about the past. So those are the bits that we can't get by, basically we can't get that from a dead brain. So this requires a living brain to get these parameters. But what we're going to try to do is to see how far we can get with dead brain data. And so now what we're going to do is we're going to say, in addition to these things that we now have some measurements for, we know what this circuit is supposed to do. The circuit's supposed to do vision. And so what we're going to do is we're going to train this network to try to perform a visual tracking task. And the idea is to see if that will allow us to infer these parameters that we didn't know. Whatever those parameters, the values of those parameters are, they have got to be good, or probably good for solving vision problems. And so let's see if you can use that trick. We'll get there. To start with, no. And that will fail, and then we'll fix it. Okay, so here's what we do. We take the circuit, which we get from the connectome, and then we add, a few neurons. It's basically a decoder. And what we're trying to say is, using the biologically sort of determined neural network as a feature space for the video clips that we're going to look at, can we use that feature space in order to track a moving object in this scene? And tracking now means that you have to predict the center of an object in pixel space in XY coordinates and its velocity. So 4 numbers, so XY position and XY velocity. So here's what we're going to do. Here's, what the video clips look like. And, these are actually downsampled to 700 pixels because that's what the eye has. This model that we built of the right eyeball has 700 ommatidia. And so the resolution isn't going to be quite as good as what you see here, but its job is to predict the center position and velocity of some dominant object. And so the problem is sort of slightly ill-posed at best, but the statistics of the scene are in there, and it's a reasonable thing to do. And our predictions aren't too far off in terms of the position. The velocity is quite a bit noisier. And part of that is also because the data set, has basically pans to track the objects. So the objects are relatively close to the center. So, but we didn't really want to solve this problem that well. We just wanted it to help us figure out what parameters would be good to sort of solve this problem. So here's what the simulation looks like. Here's the RGB channels of the video clips on a test set. And now you can look at over here, the Rs are the photoreceptor activations. And this pixel grid that you see, this hexagonal grid, those are the neurons, the activities of those neurons. And that's basically the resolution that this sensor has, is sort of this sort of level of information. Okay, so how well does it do? It does reasonably well. What does this number mean? I wouldn't worry about it too much because we don't really care. So, but what we want to do is now to test this. What we wanted to try to get out of this was detailed predictions at the level of single neurons and cell types about what this circuit actually, what the function that the circuit computes is, which means which stimuli do these neurons respond to. So one of the stimuli that people have studied a lot in the fruit fly visual system is that of motion processing. So this is a classic lab stimulus. So the idea is that there are neurons within the fruit fly visual system which are selective to bars moving in particular directions. and they respond more to bars moving in one direction than bars moving in the other direction. And those are these T4 neurons. And these are the first neurons to be able to compute motion, which is a spatiotemporal computation. And there's 4 of them, and they correspond to motion. They're selective to motion in the 4 cardinal directions. And so we can probe how this network responds to these lab stimuli and see, is this neuron motion selective or not? So here's the way that you can calculate that, quantify that. And the way is to sort of make a polar plot of how strongly that neuron fires to stimuli in different directions. What we'd like to see is something that looks like this. So here is the direction tuning of one particular neuron from Tom Clendenin's lab, where he finds that that these neurons fire more to stimuli in this direction and less to stimuli in the other direction. That's kind of what we wanted to see over here. We didn't see that. And so then we thought, well, maybe these weights that we thought we could imagine were the synapse counts, maybe they're not quite right. Let's try to also tweak the weights and see what happens. So we trained the model while initializing the weights with the connectome parameters, and then we trained it. And the network performs a little bit better, but again, that wasn't the point. But what we were after was what the tuning properties look like. And now we actually found that it did the right thing. These neurons are selective to the 4 cardinal directions. And this is really exciting to us. Also, what you see is that if the bar is moving really, really slowly, basically is almost static, that measures something called the orientation preference, and it should be kind of almost symmetric, and we kind of recover that. And so if we learn basically the threshold, the time constant, and the weights, we're able to get at this. So what happened in the network? We looked at the inputs to these T4 neurons, and this is where, we looked at what the connectome weights were, and then what the weights looked like, these filters looked like after training. And they didn't actually change very much. Maybe there's a tiny bit of, change over there, but they didn't really seem to change very much at all. We looked across the entire circuit to figure out which weights did change in order to sort of solve this problem and now, get the right tunings in the right neurons. And we discovered that, what we had was a good connectome for the on pathway, which were the inputs to the T4 neurons and their own inputs. But we didn't actually have a good connectome or any connectome for the off pathway, which is the T5 neurons and their inputs. We totally made that up. And what we discovered was that those were the weights that changed the most, but the weights that were the inputs to T4 neurons didn't actually change very much. So this was really cool to us because it sort of showed the power of this connectome and that it was sort of a stable sort of solution. But what we needed was to make up for the weights that we didn't have good information on. And this plot shows that by color coding in red, dark red, the ones where the weights before training and after training have strong correlation close to 1, and then, in blue, the ones that are less correlated. Yeah?
Just to clarify, if 2 neurons weren't connected at all,
They can't connect again. Yeah, that's right. And in fact, this wiring, which we now have access to, is totally bogus for the off pathway, and the circuit had to sort of make up something that was reasonable there. And you can look at the direction selectivity throughout the entire circuit, and what we know from physiology is that it's the T4 neurons that are direction selective, and the inputs are not. So if we make a direction selectivity index, which tells you how direction selective a neuron is based on the polar plot, we find exactly that it's T4 neurons which are, and not the other neurons. So this means that not only are we able to show that this network develops motion selectivity somewhere, but it's actually able to localize it to exactly the right neurons and make predictions that are detailed enough that you can match that to the precise physiology physiological measurements that you've made in living brains, at least for this particular computation. So that's sort of very exciting to us, because it seems like now using this combination of data, you can maybe start to make predictions. So synapse counts are a good proxy for synaptic strength if you use them as an initialization. And if you combine that with computational function, like what you imagine the circuit is supposed to do, and try to figure out, how to tweak up the parameters that you don't know, you can get some predictions about the tuning properties. And then now, we'd like to go into other sort of systems and see what we can do with this sort of line of work. Yes? Oh, yeah.
I was wondering if you tried other tasks. You were sort of saying like, you get the task close enough, but not
Yeah.
good enough, but I could easily see an argument going, you got to get the task right ultimately. So why are you sliding so easily on getting the task?
Yeah, so what we haven't done, what we don't know yet, is which tasks the circuit is going to be good at and which it's not. We tried to pick a task that would challenge the circuit in the sense of requiring that it sort of analyze the scene enough to segment objects and know how the objects go together in order to figure out where it is, but then also to be able to track it in the video clips in order to require that it do some motion computation. So now, certainly what we are working on now is to have a panel of tasks and see what tasks are relevant and which ones aren't, but we haven't done that yet. So yeah, certainly you need the right type of task, but the details of it we don't really know yet.
Even a simple manipulation of just doing X, Y, but not having to do velocity is like a subtle one.
Yeah, so
That really matters.
that's a good point. So I would guess, based on our results, at how bad we are at the velocity, that it wouldn't matter that much. But that's what, yeah.
In terms of matching the properties of the neuron when you focus on the direction,
That's right.
but there are many other say, aspects of motion sensing neurons, all the way type of effector,
Yeah, so far we haven't tried to interpret the circuit, but also and try to figure out what's going on there. What we're focusing on at this point is to add more data for the off pathway, which we currently have, and the new connectome, and try to also improve the quality of the modeling to try to get better sort of temporal predictions of the temporal properties of these neurons, to like step responses and impulses and so on. But interpretation is coming. Yeah. So I don't have anything yet. Okay, so what I told you about right so far is about how if you have the right constraints in here and you knew what the input-output sort of computation for a particular circuit might be, how you might go about trying to identify the precise nature of the neural network that's doing this computation. But there's a lot of places where we don't have all that information. And now the question is, what can we do from a bottom-up way? And so that's what this is about. So in particular, imagine that you focus in one brain region within a mouse, let's say primary visual cortex. We don't have, precise information about what the statistics of the input to the circuit might be, or indeed what its computational role is. Exactly what is the goal of one particular brain region within this visual hierarchy. I don't know how to sort of constrain that perfectly. So the bottom-up approach might be, let's see if we can measure the single neuron input-output properties, and then measure local connectivity, and then use that to identify the structure, the nature of the neural network that's in the circuit. We won't directly know what it's trying to do. Now we'll have to, once we have a simulation of this circuit, we'll have to try to study what we get out of it. So two sort of techniques that we're going to try to work with to get at this question is, basically microscopy combined with optogenetics. So single neuron computation and network sort of connectivity. We'll use calcium imaging, which allows us to record from activity within local compartments within single neurons, and then also local networks. And then we'll use optogenetics, which allows us to perturb the neural activity. But in order to sort of get at the answers, we'll need to do some analysis. And that's sort of where I come in. So a colleague at Janelia, Carl Svoboda, built this microscope, which he calls a mesoscope. And what you're seeing here is a video of neural activity in a living brain. And so these bright spots, which are blinking on and off, are single neurons. And if you pull out the brightness of those bright spots and look at the fluorescence in each of those spots, you can get these time series, which correspond to the activities of each of these neurons. If you zoom in and look at a single neuron, this is actually a neuron and a half, but let's look at this particular neuron. If you look at the fluorescence, the brightness of this neuron, this is the signal that you're measuring using your microscope. And this is the signal that you might measure if you use an electrode and measure electrically from the signal, where each of these are action potentials. Now, the neuron, these neurons in mouse visual cortex spike, and they communicate and compute using action potentials. But the measurement that we're making is not that of the action potentials. It's of these calcium transients. It's called calcium imaging because what you're looking at is the response of this neuron to spiking activity by changing the intracellular calcium concentration. And what happens is that every time there's an action potential, there's an increase in the calcium and then a slow decrease. So the readout that we are using, using this powerful technique, is an indirect measure of the actual spiking activity of the circuit. But we'll need to get to the actual spiking activity in order to be able to do science with it, to answer the questions that I alluded to earlier. So what I'll tell you about in like a quick sort of description here is the computational tools that we developed to sort of answer this question. And I'll get at, I'll talk about it in the context of this mapping, of going from a fluorescence trace to the underlying spiking activity. Like, how do we do that? This is called spike inference in computational neuroscience. And once we develop this little tool for doing this, we'll expand this and apply it to the problem of inferring connectivity and then inferring input-output functions of nodes. So there's two classic ways that people go about solving this problem. The first approach that people developed is what I will call a model-based inference method, which has pluses and minuses. And then there's newer methods that people have developed that are model-free, which also have pluses and minuses. So let me walk you through both of those techniques. So when calcium imaging first started out, people thought that we know a lot about the signals and how they're generated in the brain. So what we know is that every time there's an action potential, there's a change in the intracellular calcium concentration. And that change in intracellular calcium concentration is sensed by a calcium sensor that we've introduced into these neurons, either a genetically encoded calcium sensor like GCAMP or chemical sort of dyes. And then we measure it in a noisy manner using our microscope. So we know a lot about this transformation from spikes to fluorescence. So maybe we can use this forward model and try to invert it. So this is a statistical inference problem, a Bayesian inference problem. And one of the classic techniques that you might use for something like this is something called Markov chain image probes. So this what is in principle a nice, elegant technique, It's quite universal and powerful in what you can do with it, but it doesn't quite scale to the type of data that we're working with. This space of spikes over here is an extremely large space because the number of spikes that you can have within a particular session is quite large. And this inference, this statistical inference, where you'd like to infer not just one sort of candidate spike train, but actually the space of spike trains that are consistent with this is just not that good. So people developed these deconvolution-based methods, which are quite popular. This is a lot of this was from Liam Paninsky's lab, and there's techniques elsewhere. And the basic idea is to say, well, let's forget about the uncertainty that we have given this measurement, and let's solve this statistical problem in a much cruder fashion. You know, we know that there's spikes and they're sparse, but let's just say that they're sparse. And the spikes, well, you know, let's be continuous there. And there's a bunch of other approximations that you make which allow you to do faster sort of inference. The primary approximation is that this whole model has to be linear. And this is something that's absolutely not true for modern indicators. But it gets you a fast solution. So there's an opposite approach, this model-free, where people said, you know, this is the advent of modern machine learning, and people said, well, you know, let's just get some training data and train a classifier to detect spikes. Seems really straightforward. You know, let's build a classifier that takes windows of time and then predicts whether or not there's a spike over there. The biggest problem of that sort of technique is that you need ground truth data now. The first technique did not require that you have paired measurements, simultaneous measurements of both the fluorescence and the electrical activity. Just needed that you had a model for how you went from one space to the other. Now you actually need these measurements. These measurements are not easy to have. You know, I think, you know, it's safe to say that there's maybe only 100 such measurements you know, throughout the world in like very different settings, and there's quite a diversity of these 100 cells. I mean, so there's you know, quite a diversity from cell type to cell type, so it's not particularly constraining. So while this technique seems promising, it's not a high throughput way of sort of solving this problem. So we wanted to try to combine this model-free and model-based way and try to get something that's fast. We call this deep spike because we use deep neural networks to sort of solve this problem. You know, that's the general theme. We have neural networks everywhere. And so now we're going to use a neural network to try to analyze neural activity. And so it will have the advantages of both of these techniques. We'll use a generative model, and we'll do this completely unsupervised without any training data, which is now a new way of training neural networks. The old way of training neural networks, which is supervised learning, is you have your input, you have your desired output, and you have ground truth data, and then you just do gradient descent to try to optimize the parameters of this neural network, which we call a recognition network. In this context, in the statistical world, that's the language that people use, and this predicts the probability of spikes given fluorescence. But now we say that we don't have this ground truth data, but instead what we will do is we'll say that we have this model that can take us from spikes to fluorescence. So we're going to use that to compare our predicted fluorescence based on these spikes that we inferred and see how well we're doing. And then when we make mistakes, we're going to have to backpropagate all the way through this. And this is a challenging computational problem, credit assignment problem, because you have to now calculate gradients through this middle layer, which is stochastic and discrete binary. And so this poses particular challenges to sort of the training of neural networks. But it's been solved in recent years. And you know, we've sort of worked on this as well. And so this is the new technology that we can now use to train a spike inference network. When we do this, this is actually, we train two networks which make two different approximations. But this is our favorite, which you know makes the fewest approximations. So there's a very nonlinear dynamics that we've described based on measurements that people have made that describes how the calcium sensor, sort of how the calcium concentration changes, and then how the calcium indicator that you have binds and unbinds calcium ions, and that is sort of the forward model that we use. And then we have a neural network that now predicts that basically takes as input this green trace, and then every time I run it, it's a stochastic neural network, it'll give me a new spike train that it thinks is the correct interpretation for that data. And the cool thing about the stochastic neural network, the reason we wanted it to be stochastic, is that now every time I run it, and each of these rows is a different run of the same neural network, it produces some uncertainty. So if I compare all these runs, I'll see that I'm pretty confident that there was a spike, but I may not know exactly where it is. And that's sort of reasonable given the limitations of the indicator and the speed at which you're imaging. And that's the type of information that you want. It's something called a correlated posterior. Basically, every other method will give you something that's called a factorized posterior, which basically just tells you the local rate at each time point. And the interpretation of it looks like this, where you're not really sure even whether or not there's a spike when you try to interpret it. And so this is kind of what you would like to get at, but and now we can do this basically faster than real time, a lot faster than real time. You can quantify the accuracy, and you can quantify. Basically, we're at this point state of the art, the best. Even though now it starts to become limited by the noise in the data, how well you can do, and at the same time, we're very fast, a lot faster than real time, which these numbers indicate. But that's just sort of preamble. We developed this new way of doing Bayesian inference, which we needed to analyze calcium data. But what we want to get at is these two problems. So I'll quickly go over the connectivity inference. And so here's the idea. Imagine that you're watching neural activity in a particular brain region. You may notice the two neurons are correlated. And so now the question is, are they connected to each other? Well, using purely observational data, you can't get at causality. And that's because two neurons could be correlated either because there's a direct connection or because there's common input from another brain region. Especially if you're looking at primary visual cortex, you don't really know which of those cases is true. You can't infer causality from observational data. But now, if you do perturbation experiments, that can sort of help you sort of figure out which is the case. And that's what we're going to do with Chandler-Dobson. Unfortunately, it's going to not really solve the problem all the way, which is why you need sophisticated statistical. And the reason is that, here's the experiment that we're going to do. So we're going to have the same neurons express two different proteins, a calcium indicator, GCAM6S, and a Chandler-Dobson C1V1, which are spectrally distinct, so that allows us to perturb small populations of neurons in specialized ways. We collaborated with Michael Heuser's lab in London, and what they developed is this technique using a spatial light modulator to stimulate random subsets of neurons. So they can address individual neurons and say, whether or not they want to stimulate those neurons. And so here's 10 neurons they decided to stimulate, and every time it was stimulated, the calcium, you see calcium transients, which means there's spikes generated there. So we're going to use this technique, and we're going to set up an experiment, the protocol, where we stimulate 5 to 10 neurons, but a different subset of 5 to 10 neurons every half a second, and we'll do this for an hour. And we'll watch simultaneously the calcium sort of based activity in all the neurons that we have in our field of view. Now, what we'd like to happen is every time we stimulate one neuron, other neurons go off, and we just sort of figure out which neurons are sort of correlated to which stimulations. Unfortunately, that doesn't really work very well. Any given neuron is receiving input from lots, thousands of other neurons in the rest of the brain, and only a tiny fraction of its inputs we're actually able to observe. And each synapse is not that strong. So a single synapse isn't strong enough to create action potentials in a downstream neuron, and so these are strongly thresholded. And so it means that, you know, the effects are going to be very weak, and we need to have a very sensitive detection method in order to figure out whether this perturbation had any effect downstream, and to figure out which neurons it had an effect on downstream. And so now we're going to use this technique that we developed, these variational autoencoders, to do simultaneous inference of spikes, the external input, which, you know, we didn't get to measure, and the connectivity in this framework in order to solve this problem. And that's an inference problem, you know, people haven't been able to do before. And now we can get connection matrices from perturbed activity, which, you know, so far, you know, we don't have ground truth for this because no one's done the connectome for primary visual cortex at this scale. But, you know, statistics sort of correlate with what's well known about, you know, connection probabilities falling off with distance and correlating with signal correlations and spontaneous correlations and so on. So this seems promising. We're now working on validating these with various sort of experiments that we can do. Finally, I'll tell you very quickly about the single neuron computation. What we know is that the computational properties of a single neuron from biophysical simulations that people have done can be quite nonlinear. But whether or not it is in real life and the nature of the nonlinearity depends on the spatial and temporal distributions of synaptic inputs. So it really strongly depends on how the inputs are organized onto the dendrite of a neuron. And so we need a measurement of the spatial and temporal distribution of synaptic inputs. And so we don't yet have such measurements, and we're going to use calcium imaging to try to get at that question. So our collaborator, Carl Sabota, performed experiments where they imaged a small number of dendrites on this neuron. And so here's what the neuron looked like. These were the basal dendrites, and here's the signals, and here's the video clip of what that imaging experiment looks like. In ALM cortex, pyramidal neuron, and it's doing a decision-making task, which is indicated here. There's sampling, delay, period, and then a response, none of which will be that important to us. But the point is that this is an awake behaving mouse where they're able to do this sort of very challenging experiment. And so if you look at different parts of this neuron, you can see calcium transients, which are different from each other. So there's a spine, which is a site of excitatory inputs onto these dendrites. And you see spine activity. And then you also see somatic activity, which is the action potentials generated by that neuron. And what you'll notice is that every time there's an action potential in the neuron, and that's the white trace, the soma, there's also a corresponding flash in the spine. And that's something called the backpropagated action potential. So every time this neuron fires, the entire dendrite lights up. And so our job is now to figure out which flashes that you see in the spine are because the soma fired an action potential and which are local synaptic input, and then unmix them. And if you can do that, then you can figure out what the distribution of synaptic inputs looks like. So people have tried this, and Carl's lab has been working on this for quite some time, and they tried a whole bunch of methods and looked at a whole bunch of different simulated activity traces, and none of them worked. So this is, you know, a problem that they thought was pretty much hopeless. And then we tried our methods using these variational autoencoders, and for the first time we're able to sort of, this is in simulation because we don't have ground truth for this sort of measurement. But there was no method that even worked in simulation, and now we can show with a fairly sort of complicated simulation, we can recover the correlations between the inputs and the outputs, which is basically what you want to get at to be able to measure the input-output function of a neuron over a range of correlation values. So that's sort of very exciting. So we're now using this to try to map connectivity. So there's a few reasons why this works. We're able to model a lot of details in a much more careful way, basically for free. You know, this new inference method that uses neural networks and their autoencoders allows us to make our model as complicated as we want without hurting performance. And so that's something the previous Bayesian inference methods didn't really have the ability to do. So, you know, we use these keywords, these machine learning keywords, to solve these problems, which we're now starting to analyze the data. Yep, and so with that, I'll end. I showed you, you know, work on constraining a neural network using Connectome, derived information from, basically information from dead brains, and to try to make predictions about the tuning properties of individual neurons. And then I told you about some other approaches that we've been using from a bottom-up manner to try to constrain and figure out what the neural network in the brain looks like using, you know, information from a living brain. With that, I'll stop and thank my collaborators and take questions.
That was a great talk. If you took multiple dead connectors of the same exact structure of 5 digit system and could map the same neurons and then the average of the weights between them, do you think that might not need to make the computation?
Yeah, so I would suspect that the answer is yes, but there's a few things we were compensating for there. The biggest thing we were compensating for there was the fact that this off pathway, which is important, we didn't have the connectome score. So that's the biggest source of variation. But then, you know, any measurement that we make locally is not necessarily going to hold out perfectly across the retina. What we found locally is that the connection statistics are very similar to each other. And so that's quite heartening. But, you know, the brain isn't able to have exactly the same weights throughout the entire retina. And so what we're recovering with our model is some average version of that we're constructing, which is not the truth. And so that will also require some things. Yeah, so if you had that, which we kind of do, in this case we wanted to see how far we can get without that using an alternate approach. But certainly that will help provide constraints in places where you don't have precise information about the functioning of the circuit. So in mouse visual cortex, if we had a connectome and we had some functional sort of measurements of activity in response to particular stimuli, that's exactly the kind of thing that would certainly help quite a bit. Yeah. But here, you know, we also wanted to see if we could predict things that without using that information.
So you can predict the function, weight,
Um,
you know the conformity weights, then you can
Yeah, so we phrased function very differently in the two different, yeah. So here was the question, can we predict the tuning properties of individual neurons? And so if I put that in, then, but we could still, you know, there's a large number of things we could still do with that approach. And certainly, you know, the pushback I get, when I present this work to fly visual neuroscientists, they say, okay, great, but we already knew that these neurons were motion selective. Why do I care? We have all this rich data that we have measured. Why don't you use all of the data to constrain the model and give us the best possible model? And that's certainly valid, and we're collaborating with Axel Borst and others to sort of do exactly that. But the purpose of this particular exercise was to see how far can you get without that. Because there's other circuits where you don't quite have that.
Let's comment. So another way to use the connector fly is to the other way around to ask what are different functions that the circuit can do that are not known.
Absolutely. And so that's the way that we, yeah, that's sort of what we're working on right now, is to sort of, you know, clearly it's not just doing motion, and that's why the task that we picked, we didn't just pick motion. And so we can characterize how these neurons will respond to lots of different stimuli. and make sort of tuning curves for every neuron, which we don't really have right now. But then also ask which neurons are important for which types of computations. So if we build a decoder, once we've sort of tweaked everything up, using subsets of neurons for a panel of different tasks that we construct, either for tracking or for recognition or for, which neurons are actually important for what, which you can think of as a proxy for what the role of that neuron is. I mean, any one neuron is probably doing a lot of things. I mean, these T4 neurons are probably not just computing motion. If I train a neural network to perform a task, there's no real one role that they serve. And so, yeah. Any other questions? Yeah.
If you attempted to train it to a non-visual task, you would hope that it would fail, I suppose, because you would hope that it would be specific.
Yeah, someone just asked me that question a few days ago, and I wasn't quite sure how to answer that question. It kind of depends on how you encode the inputs and how you encode the outputs. So if I really, make it look like a visual task, if I take audio data and sort of make it look like, if I encode it, there's going to be a way in which you can encode it where it could probably solve that task. So, you'd have to specify what that looks like.
More changes to the weights, non-visual tasks, you think would be
So, but you can put all the work in the encoding. So, I kind of don't, I take this motion circuit and I can say that it's going to be a cat and dog detector. And for every time there's a cat, I'm going to say there's upward motion in the image. And downward motion for the other thing. So it's not, to my mind, yet fully. I don't know how to answer that question in a smart way. I only come up with dumb ways. Yeah. I can. Yeah.
Couldn't quite see the arrowheads in the connectome on the diagram. Are there lots of top-down connections or not?
Okay. Yes, there are some. Certainly within the circuit, there are feedback loops. So I don't know whether to call that top-down, but if you want to ask whether from central regions of the brain, whether there's inputs back, yes, there are, which are completely not modeled in our circuit right now.
Could you,
There is modulation of the circuit by that, which we're not looking at all.
could you try to make it function as a variational autoencoder and see what concerns happen?
I'd need to know more from you about what you mean by that. Maybe we can talk about that. That's, I think, a longer and more interesting conversation. Yeah. I think there's a really cool question about how you would use top-down feedback and what exactly it's doing. And, yeah.
Maybe design a more complex visual task that you will not be able to do with the prediction.
Yeah. Like digit recognition. I don't know. Yeah.
Prediction, something that's not more than velocity, not also prediction time.
Okay.
You may then be able to
Do with a longer feedback loop. Yeah. That's possible. Yeah. I don't quite, yeah. Absolutely. Yeah. We're also sneaking more neurons in that way. So, yeah. Great. If there's no further questions, thank you very much for your time.
Hello.
apply this sequence. And what task does this get?
Thanks again, Jim.
Thank you. You go back and say which task is best for you to find the right way.
Absolutely. Yeah, yeah.
There's just got big ass spaces. Just
Yeah.
like doing
Jim.
That's right. Which test is it really bad at? I mean, that's really important to know, right?
Right.
I mean, I think that, and then compare that.